very happy today to welcome everybody to the 16th webinar of the online edition of the Proton Electrostatic Conference. The speaker of today is Marilyn Gunner from City College of New York. She is very well known to all of us. Anyway, I will introduce her as all the other speakers in the series. So she has got her PhD from University of Pennsylvania, and she is now professor of physics and biophysics at the City College of New York. Among her interests, there are electron transfer and proton transfer reactions and computational estimation of pKas. The title of the presentation of today will be Transmembrane Proton Pumps, Characterizing a Protonation Microstate in Proton Loading Site and Transfer Path. So welcome, Marilyn.
Okay, well, thank you. Let me share my screen, and I kind of am excited to be here. But many of the people who have been attending this meeting are people whose opinions I really value. So I will be curious to hear what you guys have to say. I'm going to do this a little bit backwards in that I'm going to be talking first about the kind of sciencey part of the biophysics part of things, and then I'm going to at the end talk about some of the underlying techniques that we're using, because those are really new, and I'm particularly interested in this group and hearing what people have to say. So let's see. So again, for us, I want to just remind people like why I think continuum is really useful. It is physics-based. It's a coarse-grained representation of water. It equilibrates instantaneously. This is actually the frequency that you need to get to a certain dielectric constant for water. And, so you really need 100 picoseconds to get water close to 80, dielectric constant of 80. So it takes a long time to equilibrate water. And, and the water is really important for determining electrostatic energies and getting the appropriate polarization. And in MD, it's 1, and that always worries me. I think Alexei Stokobrukov has really, made me think about this in new ways. Obviously, you lose information about the distribution of waters and their hydrogen bonds, and we will in MCCE use a little bit of both. I think a lot of you know about our technique, and I think this is the second week after Tom's talk last week. We're going to be using Monte Carlo. And I just want to, so we have basically a rigid backbone. We can have heavy atom rotamers. We don't always use them. We always have full proton. So, we'd always have, say, the serine with a carbon here, or rather with the oxygen here, having all these different proton degrees of freedom. We can do full rotamer search. When we have explicit waters, they have lots of hydrogen bond positions. Every acid and base can change protonation state. We can do small molecules, which can be, say, a chloride, or if we are doing explicit waters, they can leave the protein. We do redox states, but we do have a rigid backbone. So I'm going to talk for maybe 10, maybe 15 minutes about proton pumps, and which is what we do a lot of thinking about now. I'm going to start with what we call complex 4, cytochrome C oxidase, which is the terminal electron acceptor. Electrons are put on oxygen and make water and pump protons. And when you get no oxygen, this is why you pass out and die, as this guy needs to keep the electrons moving down your mitochondrial electron transfer chain. And I'm going to talk a little bit about complex 1 or Nadh dehydrogenase near the end. A pump kind of is a complicated, lovely little machine. It's going to take protons from the low concentration side and dump them on the high concentration side. In order to do that, it needs 2 things. It needs gates, at least 2 gates, so that the protons come in when their gate is open to the low concentration side, and then the gate is open to the high concentration side. So we're going to need to block things at, have blocks in the proton transfer. And proton transfer, I will assume, will be mostly via the Grotius mechanism. The other thing that, and in fact, this is something MCCE does pretty well, is that when the low concentration site is connected and open, there's a proton loading site. There are amino acids or residues inside of the protein that have a high proton affinity. And then what happens is the reaction goes another step down the reaction pathway, and you've got this bait and switch, because now the proton affinity is low and you are open to the outside. And this is how you can thermodynamically drive, say, come on in, and the water's great, and oh my God, no, you want to leave. So that's as long as the gates and the proton affinity are synchronized, you're cool. So there are proton transfer pathways that are hydrogen bonded networks that use GROTUS. There are gates that open and close, and very often dehydration events that remove waters. And there are proton loading sites that change their affinity. And in our lab, we use molecular dynamics to create, or we've actually done a lot of this with QC and with other people, with Abi Singara, to get molecular dynamics trajectories, although we do our own now. And so that samples conformational space with fixed protonation and redox states. And then we use MCCE, which lets us find the protonation, equilibrium protonation states, and also the equilibrium hydrogen bond states that are consistent with the backbone that we got from either molecular dynamics or from a crystal structure. And then we use network analysis to find the proton transfer pathways. So in cytochrome C oxidase, you see you have like a connected chain of proton transfer guys. You have a lot of ionizable residues, polar and ionizable residues on the surface. And here you have what I'm going to describe in a few minutes as a complex cluster proton loading site, just a tangle. And so what will happen here is that the electrons will come in from this side, and you'll essentially bring in 4 electrons. From this side, you'll bring in 8 protons from the bottom, from the low concentration side, the N side. 4 of them will do chemistry, and 4 of them will be pumped out. And so we want to figure out where the protons get stopped and where the gates are. So what we see is that this is this network analysis, and each one of these dots is a residue. It's usually an amino acid, but can be a propionic acid on the hemes. Each line is a connection between 2 residues that can be either direct, but also can be through 1, 2, or 3, or 4 waters. So what you see here is this is the famous D pathway. It's got an aspartic 132 at the bottom. Here's a bunch of polar and water residues, no ionizable residues. And then here's another ionizable residue, which is glue 286, which we're going to talk about. This mare's nest here kind of separates into a very tightly entangled group of ionizable and polar residues, another group that's a little bit less entangled, and then goes out to the outside via, again, entangled clusters, highly interacting, and multiple pathways here, multiple pathways in. And so we would say this is a complex pathway with a cluster proton loading site. This is a relatively linear pathway with single site proton loading sites. This is a single glutamic acid, single aspartic acid. In this, there's probably there's 8 or 9 ionizable residues, all of which are coupled together. And so I want to just show you how this is kind of would not really be very good for the protein, because a proton can come in from the high concentration side and go all the way through. So you need a gate. And so there's a glue cavity that we found with QC doing molecular dynamics. Pooja Gowal, who's now at University at Binghamton, did this. And In the crystal structure, this is closed, and basically there's a break in the hydrogen bond network. So this is on the high concentration P side. This is on the low concentration N side, and there's a break, and that's that. And this is what you'll see in the crystal structures. When we put a proton about 15 angstroms away from that glue, on that propionic acid, suddenly a hydrogen bond breaks up here, water comes in, and now you see connection. So this is a proposed gate, and I've tried to do some experiments to find it, but have not had much luck yet. This is, again, the closed system, and that glutamic acid is in a very dry neighborhood, not near any other ionizable or polar amino acids, and it holds on to its proton very strongly. These are a bunch of different redox states of the protein. But when you open the gate and you want to be able to lose the proton to go out to the P side, the high concentration side, when the water comes in, the hydration of the glutamic acid makes it much more happy to be a negative charge, and essentially the proton affinity goes down and you can release a proton. So this is a simple proton loading site, a single residue. It exits to a linear path with basically one entrance, one exit, and hydration opens and closes this. And it's a trigger for opening and closing is 15 angstroms away. Okay. So now I want to talk a little bit about a cluster of proton loading site and a complex proton path. So this is, let's look at this. This is actually a slightly different cytochrome oxidase, but very similar. And it's basically got 6 residues. So Xiaoheng figured out that there were 4 amino acids and 2 propionic acids that are tightly coupled. And instead of having one residue like Glu-286 that's either ionized or neutral, now you have 6 residues, which give you at least 64 microstates of protonation microstates. You're not going to use them all, but you have lots of possibilities for where the proton's going to be. And what she did was, this is a little complicated, but let me just say, so in those 6 residues, they go between a charge state of -4 to a charge state of -3. So when it's unloaded, there's 1 proton in the cluster. And when it's loaded, when it's got its the proton loading site has loaded a proton, it's got 2 protons distributed amongst those 6 amino acids. And each one of these is for one underlying structure. So this is one PDB file or one MD snapshot. And so we're going to look at the purple thing because that's bold. So that's where I want you to look at. And in the starting case for this particular structure, the proton loading site has a charge of -4. It's got 1 proton in it when it's got an electron on this heme. And when we move the electron from one heme to the other heme, this loading site, we didn't tell it, but these 6 residues now kind of begin to pull up a proton. So about 6, on average, they're going to bind 6/10 of a proton. That means some of them bind 1 proton, some of them bind no protons. On average, it's 0.6. Okay? Now what we're going to do is the next step in the reaction cycle, and we move a proton into the chemistry site. And when we do that, again, we didn't tell the protons what to do here, but these protons on these 6 amino acids now go away, and we go back to just having 1 in the cluster. And then as we put another electron on this heme, we don't see any protons coming in. And again, we're going to need to do 4 electrons and 4 protons. So each one of these up and down here has a somewhat different redox state in the active site. So again, we don't really care, but at each time we basically move the proton from heme A to heme A3, we see loading. Each time we put a proton on the substrate water in here, we see unloading. Now that's good. This is dynamic. We did not tell the program which were the 6 sites, so the program told us which were the 6 residues. So the Monte Carlo analysis lets us see complexity, lets us find not a single residue that's the proton loading site, but a group of residues. Okay, all cool. But what I want to do is now look at why this protein held on to its proton for, didn't ever give it up. And then we had another protein down here which never bound a proton. So what's going on? So we have some that are trap loaded, some instances of structures trap loaded, some trapped unloaded, and some structures that are dynamic. And that's going to tell us something interesting about, tell us something interesting. And so when you have one proton among 6 sites, you have 6 potential tautomers. And we're beginning to see that I think proteins are using tautomers to do a lot of work. And so when you have basically the proton tautomer with only one proton on this aspartic acid, then it's gonna be ready to bind another proton. But when the protein structure is stabilized with the proton on this propionic acid, then it's overstabilized. And I'm going to say this again in a minute. So let me just, and again, when we have the loaded structure, 2 protons, if it's on these 2 protons, then it's ready to lose the proton, gain the proton. But when it's in these 2 places, it's trapped. So let me show you what this means. So this is a Tautomer trap. So when I have the proton in these 2 places, it's largely protonated. Okay, so we're largely loaded. And if this is pH 7, we're loaded, but the proton affinity, and again, I'm drawing these as just simple Henderson-Hasselbalch. They will not have this cluster doesn't have that kind of behavior, but for us, I'm going to do that. So basically in the loaded state, it's loaded, not 100% loaded, maybe 80% loaded. And the proton affinity, the effective pK is a little bit above the pH. And now when you do something in the active site and you make it want to give up its proton, again, you're not going 100% to give it up. You have a modest shift in proton affinity, and the proton, has basically left one of these sites, okay? And so this is an active site, and it's good in one sense because it can respond to the protein. It's bad in another sense because it's not binding stoichiometrically. So we can also have a different thing where we have these very far apart, very highly favored. Even when you want to change the proton affinity, it doesn't change the proton loading. Or you could have it so that the unloaded state's very low in energy. Even if you change the affinity, you never get to you never load. And so by moving from being in this state where you have very low affinity with one particular tautomer to another state where the affinity is moderate, to another state where the affinity, again, is very high, you can basically go from a trapped unloaded to a dynamic loaded unloaded state to a fixed unloaded state. And Xiaohang did this on like 100 structures, and we see a group of structures that stay in the loaded state in loading conditions and unloading conditions. They stay in the same state. Another group that stay loaded, and then they move down to have a charge from a charge of -3 to a charge of -4 as you change the conditions in MCCE. Again, I don't need to show you this, just to show you that there's about 100 structures underneath this. And we can start to see that the trap state have a particular hydrogen bond pattern. The dynamic states have a different hydrogen bond pattern. And so the trap state has a the trapped unloaded state has a very large distance between these residues, and when they're trap loaded, they don't make a hydrogen bond, but they get closer. So this is, I think, Tautomer trap is something that can be seen in a complicated PLS, where you have multiple residues. Here, small changes in the conformation can ensure states where, move you between states where change in protonation is easy, to change in, and they can relax into trap states where basically it's harder to change the protonation state. And we move from the trap to the dynamic states to allow, we think, or we propose that the trap states kind of fix you there for the perhaps milliseconds while the gates open and close. Okay, so that was a bit heavy. I hope that was clear enough. But now I want to talk about the underlying microstates and just where we're pushing MCCE to be able to work with these microstates very simply. Okay, so what I want to do is, this is a new methodology that we're doing, and I would say Junjun Mao, who's been working on MCCE since a long time, is, with Yifan Song in the beginning, and has just been hanging out with us more in the last 5 years or so and really gone a long way. And Umesh Kayana has been doing the microstate analysis on lysozyme and on complex 1. And let me show you this. Okay, so we have in Monte Carlo, we have in MCCE, we pre-make the conformers. It makes it very efficient, and is going to make this way of saving microstates again efficient. I don't think you need to do it this way, but this is how we've done it. We, in a standard MCCE calculation, will have from 5,000 to 20,000 conformers. Again, I think we can push this up now using, more, more, just being a little cleverer. So each one of these is one conformer. So each residue might have from one conformer for an alanine to hundreds of conformers for waters or an arginine or an aspartic acid. Explicit waters are removed when we calculate the PLS protonation states. I think it's just more efficient, and I actually think it's probably more accurate, but we go back and forth. But they're retained for hydrogen bond networks. So actually for most of the problems now, we will be doing runs with both, and we can compare the protonation states and other things. And we make tens of millions of microstates. So each microstate is like each step in a molecular dynamics simulation. And we've actually now can replay the full Monte Carlo simulation. So we have a very compact text readable file that has the information of each one of these tens of millions of steps. And we can then generate access to all microstates. You can't write out all the microstates at one time, but you have access to it. And if you ask focused questions, you can interrogate the data set. So I'm going to talk a little bit about lysozyme. So
excuse me,
yes.
Marilyn. Yeah, this is Steve. Can I just ask just a really quick question? So
yeah.
All these millions of microstates, are they genuinely independent or are they correlated in some way?
Well, I mean, in Monte Carlo, there is some memory because you only make a certain number of steps. We will change from 1 to 10 residues at a time. But there's we expect no history over the long run. And that is, in fact, what we want to see is what the correlation is. I mean, if we only cared about averages, we didn't care about correlation, then we wouldn't care so much about microstates. So that's where we're really going to start seeing correlation.
I see.
Does that answer your question?
Yeah, thank
okay.
you.
Yeah, so any other questions? Okay, so this is for lysozyme, and this had, 3 million total microstates, about 200,000 unique ones. And when we get to bigger proteins, more of them, we have larger percentage, I think, are unique. Because again, and so the enthalpy actually there's a fair amount of entropy in MCCE calculations. I mean, the lowest energy states are down here, and we have a range of 25 kilocalories. So So I think one of the things we've been thinking about is if we want to give, say, molecular dynamics the right charge states, like, or do we want the lowest enthalpy state, the highest occupancy state, what do we want? But let me just tell you a little bit about what we're seeing. And so this is, again, lysozyme, very simple, and here are some amino acids that are titrating in the range of interest, which is, I guess, from about 4 to 7. And what we see is, again, this is the count. These are modestly sized bins. You can certainly bin these. And so this is the number of unique microstates in each enthalpy bucket. Okay, and then this is the if you want to actually get the Boltzmann average, you need to count each unique state by how many times you had to accept it before you found the next unique state that satisfied the metropolis sampling. So these look pretty similar. They're not exactly the same. So we actually, for this, we're doing a lot with just unique states, not with the unique times the count, but we go back and forth. So this is pretty, I think this is kind of interesting. This is, again, lysozyme pH7. And you see that we see, and I know Tom Lowry's gonna say, well, this is just the amino acid charge, not the ion cloud. But this is, so there are states with charges of 0, -1, -2, -3, and -4. Actually, then you have to add 10 because we didn't add. So there's, okay, slow down. There's 7 aspartic acids. 4 of them are changing their charge in the accepted microstates. And 3 of them basically are always in a single charge state. Again, glue, CTR, so of course all the arginines are in a single state. So again, and so we have 10 amino acids that are changing, and we have essentially a background charge of 10 that wasn't included in here. But so what we see is that we see charge states from 0 to -4, which is really from 10 to -6, if you care. And they range over quite a large enthalpy range. So this can be in this, and this can be several different tautomers in this line. These are many, many states. Or they can be the same tautomer, but have different serine positions or different conformations. Okay, and you see that the lowest enthalpy states have a charge of -2 or -3. And this is now the unique protonation microstates. So this had, as I said, many conformational microstates in addition to charge. So at pH 7, you have 27 unique protonation microstates. You have a few that are fairly, so this is a log of the counts. This is, you get 1,000,000, 100,000, and then ones that are maybe only seen once or twice. These are the charge states. So these are now different tautomers of each of these charge states. And these are the ones that are going to be in the low energy Boltzmann population. And these are ones that might be interesting for other reasons. And again, just to remind you, for a given number of sites and given number of protons, M, you have these number of tautomer states. Okay, so that's the rule. And so this is the average protonation state in the Monte Carlo average for, say, 6 amino acids that are changing their charge at pH 7 in this calculation. So we have 6 residues. It could be 64 possible microstates, but only 27 of them are in the Boltzmann, in the 20 million Boltzmann accepted states. I suppose we could keep going and see more of them. If we look at the 2 most occupied states, we see that, okay, well, this is 14%, and these are both 0. This is 36%, and you see one microstate with this being neutral and another with being protonated. These are all, boring. And so, these are the 2 most occupied. And this is actually the relative count in this lowest enthalpy bucket. So you look at the lowest enthalpy bucket, you see that this one, so that's there. And you can go through, these are all of the 27 different charge states. So this guy's only seen 7 times. This one's seen 600,000 times. So if you ask me now, what is the right thing I should do to say to put in a molecular dynamics simulation, I would say, well, this one or this one's probably pretty good. This one might be interesting. And so even though we see many, it's not completely intractable. And so we can again look at them. If we go down to pH4, where there's more going on, we see now 200 unique microstates. We see more in this higher population. This guy here has 15 kilocalorie range. So this one unique charge state is found with basically conformational microstates that range over 15 kilocalories. So it's got it's a big high entropy bucket. This one has, slightly smaller. And I guess as you get down to less occupied, they tend to have a smaller enthalpy bucket. We just didn't know what we were going to see here. So at pH 4, where all the asps and glues are titrating, we certainly see more occupied microstates. At pH 7, where there's only a few groups in lysozyme that are titrating, we only see 20 odd that are titrating. So again, we have now real control over them, and we can say, what's going on with each of these, what are the side chains around them, what's the hydrogen bond network with this particular unique microstate, or this tautomer, or this tautomer, or this tautomer. And so we can do it, we can see everything. And so again, I'm going to skip this. This is going through lysozyme because I want to spend the last minute or two. Oh, so this is perhaps thinking about correlation. So this is something that we really do want to do. And I'm not at all sure what the right way to do it. So Umesh did this with the Pearson correlation. I think we might, move on to this, which is, in the Monte Carlo calculation for ionization states, things are either ionized or neutral. There isn't a number line for ionization. You're either yes or no. So this is something which is more appropriate for a binary distribution. And so we're kind of looking at this. We don't see lots of big numbers, but here's AS52 and GLU35, which for those of us who have done lysozyme in the past, we know is correlated. AS101 is correlated with GLU7. And this is at pH 6. And then if you do this at pH 7, you begin to see other correlations creep in. So a lysine, which was, I'm sure, completely ionized at pH 6, is now beginning to become slightly deprotonated here. And it is actually correlated with this. And so we now can begin to see not just single average, but microstate correlations of ionization within the microstates. And as I said, I'm hoping we'll see, look at correlations of hydrogen bonds with ionization states and other kinds of things. Okay, so I'm going to take a breath for a moment, and then I will see if anybody has anything. Okay. And now I want to show you how this can blow up on us and where we're going. So complex 1 is a big protein in the mitochondrial electron transfer chain, and it's got 4 proton transfer pathways. And Umesh and Abhi Singharan have worked on this one. It's called the E channel, and I'm going to show you a little bit about how we're trying to figure out what to do with the microstate analysis in this, but let me just show you where we're going. So this is published. This is the network analysis of the proton transfer pathway, God help us, from the N side, the high concentration side, which is where the quinone is, where all the electrochemistry is happening, through this extremely complicated multiple, there's 200 ionizable residues in just this part of the protein, and in turn we get about 75 of them on this connected pathway. And so this is really where you need network analysis and you need Monte Carlo. I mean, otherwise you're really making ad hoc decisions about which amino acids are on the pathway. And I think when we do the network analysis, we really can see this. And this complexity is from a single snapshot from a molecular dynamics trajectory. And so it's not even adding multiple trajectories together. It's doing one Monte Carlo simulation. The side chains cannot move their heavy atoms. They can just move their polar groups, and the waters can move, and you still get just an enormous complexity of connections amongst groups of amino acids. And just to remind you, one line is basically water. It could be 0 to 4 waters between things, and reds are acids, blues are bases, and greens are polar. Okay, so we've divided this into clusters, and there's actually a break between these clusters, and that's something we're thinking about for the proton transfer network. But what I want to talk about is, what are the proton loading behaviors? We assume that protons are going to be bound into one or more of these clusters, pass through this cluster, and then come into this cluster and get exuded. So we expect to see that there's going to be loaded or unloaded protonation states. Now in cytochrome oxidase, which was a much simpler system, we could really say, oh, here's one cluster and it's the one that's loading and unloading, and that's that. This is not going to be that simple. And so again, what we have in this part of the calculation, again, these are our enthalpy things, is we had 3 million microstates. We had 80,000 unique charge microstates, just unique charge microstates. And, so that's really intractable. But when we start doing a little bit of dividing and conquering, we have something that I think we're going to be able to use, which is if we think about, say, cluster 1 has 7 ionizable protonatable residues in it. And 4 of them don't change their charge. So 3 of them are changing. And that could give us 8 microstates, 2 to the 3. And so we see 5 individual microstates for cluster 1, not 8. And 2 of them are populated. So 3 of them are high energy and 2 of them are populated. If we're in cluster 2 with 11 charge residues, we see 6 of them are changing. We see cluster 2 has 45 microstates. So it's got a lot of degrees of freedom, but maybe we can say 3 of them are at low energy. And again, so we can see for each of them, cluster 2 and 4 have a lot of microstates that are occupied, but modest numbers at low energy and highly occupied, low free energy. This is interesting because this has got a lot of residues in it, but it ends up only having 4 occupied microstates. So in the end, we have 73 charge residues in the network. But only 24 of them are changing protonation at pH 7. And on the populated microstate, what I did was I don't know how many are populated, but I just multiplied 2 times 3 times. So I just kind of, did the combinatorics. I did not do the combinatorics here because it scared me. But it's probably a bigger number than I want. So that says that I probably have at least 100 occupied. Well, I don't know, probably too many occupied microstates. And again, we can see there are correlations. This is another thing we're looking at, is what are the correlations between them. So, what we really want to do is see that there's a loaded microstate and an unloaded microstate, that as we change the outside conditions of this trajectory of the snapshot, we see some states where one of these is got an extra proton, and in another case it doesn't. And we were looking for snapshots where this one has an extra proton and where it doesn't, and then we're going to look to see where that proton resides. The tricky part here is going to be moving back and forth between the molecular dynamics and the Monte Carlo so that we put in the right charge states in the molecular dynamics to enlighten us and not lead us down a rabbit hole. So I want to just say this is a new thing, this mining of the microstates. It's just more than we expected, even though I should have known this. We can mine correlation. We need this information for finding the hydrogen bonds. If you guys are interested, we have a new version. Junjun has really been working on this for the past year. I think it's great. It will go, it's Conda accessible, or you can go on your PC, on your Mac, on Linux. It's got Python tools. It's got a website. If you build Python tools for it, we would love to have them. We'd love to see, we'd love to get feedback on it. We're writing up the papers to go around it. And I want to thank everybody who helped, of course, Microstate, Xiaoheng, Lumesh, Junjun, Oxidase, QC's lab for molecular dynamics, and Xiaoheng and Janshen, MCCE. I have to thank Yifan and Emil for a million years ago, and current members and recent alums. So anyway, Chitrak and Abi for Complex One. All right. So I hope I have some questions or comments.
Thank you very much, Marilyn. I see a question on the chat from Diane.
Yeah, Walter, you can just read it, but Marilyn, it pertains to the very first part of your talk.
Okay.
So for mitocomplex, you describe a proton transfer from to p side. The same principle for bidirectional reverse transfer.
So some of these will do reverse transfer, and some of these, most of these don't leak very much. So complex one will pump protons backwards, and it's got to have some. So if the energy gets high enough, they will.
Marilyn, if the proton gradient is reversed for some mechanism, aren't you going to have the reverse transfer?
Basically, these are really highly. So you can push protons up about 3 pH units or 180 millivolts. And so 180 millivolts, you'll be fine. If you start getting more than that, then these have different behaviors. So in cytochrome oxidase, you actually start moving protons back from the top to do the chemistry, just so that you don't you don't want to build up, let's see, where is that? You basically don't want, you need to kind of keep the rest of the pathway going. So if this proton pathway gets too hard, then you basically just start leaking protons in from the wrong side and just keep the electrons going from the other things. I actually think that complex one can, I don't know if it will reduce quinone if the proton, if the delta psi across the membrane or the proton gradient gets too big, but that's what you'd have to look for. But all of these, once the proton gradient gets too high, they tend to stop, and then you begin to get the electron transfers backed up, and then you get reactive oxygen species. So I'd say they're poorly reversible. Why, I don't know.
There is a question from Tom Lowe. A related question is, how does the external dipole moment from the membrane asymmetry impact the proton pump model?
Yeah, that is a good question. And, you know, I mean, our calculations are all done under free load. So they're done in a sense without the membrane potential. A few people, Paul Barossa and Matthias Ullmann and others have, and I think maybe Benoit have done these calculations under load. So you basically put a gradient over here, and I guess this comes back to Diane's question, and you then should see that the energy that, you know, we're looking at when we basically, you know, put put a charge here, the protons can get bound up in this region. Well, if I put enough of a positive charge on this side, that's going to shift the proton affinity of this group because of the positive charge on this side, and that's going to lower their ability to bind the proton. And I mean, I'm really, so that people have tried that, and that's what should happen. I think it's, I don't know that it needs to be more complicated than that, but it probably is.
Okay. There is a question from Jim.
Hi, Marilyn. Well, I missed most of your talk, so but it doesn't really matter because my question would be stupid anyway. I'm a bit, so when any of us are doing these PK calculations, we have everything, so scale it up and down in terms of the the huge PK shifts, low PK shifts, all these discussions over the years. I'm thinking I am just seeing the last part of your talk surprised, as perhaps you were, by the large number of different protonation states. And and we want everything, more about membrane proteins than I do. We want everything to be sort of like, as you say, on-off sort of thing, or like potassium channel where it's just knocked out. It's got sort of 2 or 3 different states. But is there a parameter? How confident can any of us be in our methodology where we are scaling those interactions between charges? About if we scale them all to be very low, I guess we separate the energies of the different microstates to be more apart, whereas if we've got them too coupled, then they somehow paradoxically, you see what I mean? I mean, how can we be confident about the scaling that gives you the different microstates?
Well, I think, one thing is, if you have, say, no scaling, and you have all your aspartic acids, all of your aspartic acids being protonated at the same pH, you'd have maximal entropy at that. You'd have maximal number of microstates, even though they'd be completely uncorrelated. So they wouldn't care about one another, but you'd have the most microstates, because at pH 3.8, everybody'd be half charged. And if you have 10, so you have 2 questions. I mean, one is, if you start moving the protonation states so that they're shifted and that they're correlated with each other, I don't know that makes it more complex. I think, in fact, it probably makes the number of microstates smaller, because you don't have both aspartic acids ionized, you have one or the other, whereas if they're uncorrelated, you have more. So I think, because we don't have the full 2 to the n, we have some subset of 2 to the n, some smaller subset of 2 to the n, a half or a quarter. I mean, I think some of that's due to the fact that we have strong interactions. So that's a different question. And how good is our PKs? I don't know, for lysozyme, we can look at how close we are to the experimental values. Umesh has done a really nice job of looking at, a handful of crystal structures. And so, again, you always average over, look at a lot of different structures, look at a lot of different, and then begin to see, in a lot of different structures, are the same microstates showing up at the bottom. I think that's how we're going to approach this. I mean, we really have only had this data for just a few months, so we're really trying to feel our way through how to you know, how to deal with like 80,000 microstates in a way that doesn't make your head explode.
So, does it have to be a membrane? So to get these wonderful situations that all of us have been looking for years, where we don't have just an aspirin, a glue, and a lysozyme, we've got all, you know, we've got a cluster of 5 or 10 charges flipping up and down and doing something, a bit like Diane sort of stuff.
Right.
But does it have to be a membrane protein, or do you think other proteins can do this?
No, I'm sure that Diane's proteins are really primed for this, because they've got this, you know, they've got that huge, that tiny, narrow collective behavior. So in a sense, I think that she's going to see, when we can look at her proteins better, we're going to see some really interesting correlated behavior. Or, you know, kinase proteins, where you have some trigger, you know, so suddenly you put a phosphate somewhere, and, you know, something happened, you know, like several things are now feeling the pressure of that, have their pK shifted in a way that moves to an unstable tautomer, and then you probably get some conformational gain. So, I mean, this is the other thing, is to move between the conformational gating and the tautomer gating. I mean, we're now kind of moving by hand between, we think this tautomer group, so when we run with the trap tautomers, that gives us back molecular dynamics snapshots that are trapped. If we run with the dynamic tautomer positions, the MD snapshots tend to be able to be more responsive when we put them in it. I mean, so this is, I think, the thing that we really need, maybe better constant PHMD, where we can, you know, I mean, it's really scary, because as we put each one of these different charge states into MD, we dig a well. So that, to me, is the thing that I'm most.
Marilyn, this is Diane. Related to that, though, I may have missed this, but does that, on the right side schematic, does that lysine have a downshifted pKa?
Very much. It actually holds on to its proton for dear life. So it is, yes, so it is neutral but then it's going to pick up a proton dynamically and pass it on. So so it's like what we would call a single site proton loading site.
After Diane is finished, I have a related question. I don't know if you finished, Diane.
Go ahead.
Yeah, mine is more simple-minded. I liked your schematic of the, you know, simplified schematic of a pump. And you explained you need to have 2 gates. So my question is, can the gate be just an electron? Does it have to be a conformational change, or can, say, an electron move to a site close to a proton and trap it and serve as a gate? Would that be one of the possible gates? Yes,
for sure.
Thanks.
So that you can change the proton affinity of a nearby residue by 10 pH. What you really need is, I mean, to go from being 99% ionized to 99%, you know, not ionized, you need 5 kilocalories. You know, you need to be 2 pH units above to 2 pH units below. And so you need a big shift so the gate, I mean, the gate can be the proton loading site affinity change, or it can be a dehydration event.
Okay, thanks.
There is a question from Antonio. Hopefully. Okay.
Hi. Thank you, Marilyn, for the really great talk. I have several questions or comments or whatever. Well, first of all, I'm glad that you are looking at correlations, because, I mean, we started to look at that 20 years ago, and apparently no one was paying any attention to correlations until recently. So
I still don't think anybody else is paying attention to correlations either. I'm waiting for somebody smarter than me to do correlations.
Yeah, I really like your analysis. currently we are also looking at this kind of things in some detail, as you could, I mean, see probably next week. I don't know.
Oh, cool.
When Walter will schedule me.
Yes, you are scheduled on the next week. Yes.
Next week. Okay.
Yes.
Yeah. So about the correlations, I was curious. Do you see any long-range correlations? I don't. Maybe you remember, Marilyn.
Yeah.
Telluride, when I showed some results from cytochrome C oxidase, I had this really weird case of very long distance correlations between sites. Do you see anything like this?
Yes, we do.
Yeah.
And we see, I mean, the other thing that we see, and I don't know if I see it here, I mean, we see things that are weirdly, kind of anti-correlated. So,
yep.
Where you have two acids that are not, or positively, so yeah, we do. In these clusters, it's really complicated. And also, I mean, the other thing that I think we're going to have to deal with is that these are not really strong correlations. They're kind of embers of correlations rather than very strong ones.
Actually, I would say that the values you have, I mean, for this kind of, we tend to look at correlations. We are too biased from using correlations as correlation coefficients in linear regressions. But I would say that 0.3 or 0.4 or 0.5 is quite something for the kind of phenomena you're looking at.
Right.
I mean, and in some cases you can have, in our hands, we in some cases we got even higher correlations. For instance, 2 years ago, we did some constant pH MD with PSBS, this small protein from the photosystem 2 with Nicoletta Liguori.
Oh, right, yeah.
Actually, Nicoletta is here today, yeah. And in some cases, we were even above 0.5 or almost 1 in some cases. So, yeah, and this was a membrane protein. So if you have a low diuretic medium, I would guess that you could get even higher things. And the reason I think this is, I mean, the fact that you see these also in your case, I see these correlations are really interesting because they may kind of be the explanation for pH sensing in many cases. You can sense a change from a completely different part of the protein on the other side of the protein from a protonation change in one position. Anyway, but I'm digressing. Sorry.
No.
So,
I mean, I like this one because you can kind of see these are all acids, and you can kind of see the Barber pole, negative, positive, negative, so you're seeing even amongst groups of the same charge.
Yeah. Another question I have, you mentioned the trapped states. My question is, when you have more correlated residues, do they tend to be trapped?
Ooh.
They do or not? You don't know. Okay.
I don't know. I don't.
That's what we see. Okay.
Because we haven't done when we did that, we didn't have all the kind of all the underlying programs when we were doing that, and that's a really good question.
Okay. Okay.
Yeah.
Yeah. Yeah. Thanks. Well, just one. This is more a comment about the entropies, because you talked about a lot of entropies.
Yeah.
Today. I think it's an overlooked fact that even if you don't use conformational effects like you do, I mean, in your case,
right,
or in constant pH MD and so on, even if you don't include conformational aspects, I mean, you always have an entropic factor, and sometimes I think people forget that. I remember once computing 20 years ago again. I'm getting old. The contribution of the occupational entropies of the surrounding sites for a given pKa, And in some cases, it goes up to half unit.
Right.
So it's interesting that you mention entropies because people keep forgetting about them, even for the occupation of the proton sites. So,
I mean, I was just shocked though when we saw this. I mean, I got, poor Umesh and said, we're getting the lowest enthalpy states, and they're really not like the average. And I was going, there's got to be something wrong. There's got to be something wrong. And then when we finally saw this, I mean, this is a lot of kilocalories.
Yeah.
And this is even more kilocalories. I mean, so in the beginning, I just was sure that there was an error in the calculations because the average and the low enthalpy states were so different. And I think that again, this is really interesting for what we do. One of the things I think I want to do with this is say, okay, these are the best microstates for molecular dynamics. And I'd like to hear from people who need them. Do you want the lowest enthalpy state? Probably not. I mean, you probably want something here.
Yeah, exactly.
And there's still 40 of them. I mean, Umesh is looking at 10 to the a slice of 10 to the minus 4 kilocalories, and you still get 40 of these. So anyway, I mean, it's really going to be interesting now that we have access to this, what we want. But please, if people try, I'd love to have people try this out.
If you want to start a molecular dynamics simulation, a fixed protonation molecular dynamics simulation, you will need one structure.
I know.
So,
but which one?
Yeah, but if you have an ensemble of structure, then you will end up having an ensemble of trajectories, which makes sense. Absolutely.
Well, that's what our constant pH friends are trying to do. But I think that I hadn't really understood how complicated, how that was just not a well-defined question. Like to get, we think, oh, we'll give you the right microstate rather than looking at the averages and just take that. Now we know that, when two things are 0 and 1, they have a mixture, which is the right. But, I think I suspect we'll print out for people the lowest enthalpy microstate and then, several of the highest occupied microstates, 3 to, and then let people decide what they would want to do.
Yeah.
But they would be complete for the whole protein.
If I may make one more comment, I think one of the things your work also illustrates very well is the amount of microstates that are actually populated is absolutely huge.
Yeah.
And that must necessarily be a limitation in constant pH MD.
Oh, yeah. Because
the method is slower. We cannot, not in our dreams to sample protonations, as many times as you can do in your method. So this is also instructive in that regard.
Yeah. No, I really assumed for many years that constant pH MD would just, MCCE would go away and we'd have but I see that that's not. Now, we can certainly take one, I'd like to do, say, constant pH MD on the 6 residues in that cluster I showed you. That's very tractable. But I couldn't imagine how to do it here.
Yeah. Actually, one of the reasons, for instance, in our method, sorry, I'm monopolizing this, but the way we do it when we select new protonation states, we do a full Monte Carlo calculation precisely to scramble as much as possible the microstates, so to not get stuck. So yeah. Anyway, thank you. This was really great. Yeah, really enjoyed it.
Yeah, I'm gonna jump in.
Oh, hey, Anna.
Hi, I came in late.
That's all right.
My class. Yeah, so I totally agree with you on constant pH doing on like clusters of residues. Yeah, that's absolutely what we should be doing. Yeah.
maybe I will call one of you, somebody, I do want to do that on some of this, but but I think to do these large numbers is just, we need to think really differently.
Yeah, but I think the worrying part of it is that, so I would say the vast majority of people who are doing molecular dynamics, they pay some attention to the initial protonation state, and then they do not care anymore.
Right.
And if the protein is using, as Diane was showing in the previous talk,
Right.
The change in protonation as a mechanism to do something, if we cannot jump from one of those microstates, we will be blind, and we will be never able to observe something, and maybe we blame something else, some other limitation for this. Yeah. This is worrying.
I mean, this is, I think, really worrying for me as well, because when we think, I mean, even though we may run in multiple charge states, and we're getting our charge states out of this kind of analysis. So I think we are now in a position to get the best charge states, anybody can get. We're doing a good job. But each time we put a charge state into MD, we dig a hole, and and then you bias towards that charge state. And so I think this is a real difficult problem. I mean, whether we need to do some coarse graining, or whether we need, how we need to kind of add some loft to the calculation, so that we, maybe we can sample better, even if the energies aren't as precise. I think some of the ways that people are going with constant pHMD to try to, get the energies and the thermodynamics exactly right are so slow that you can't do the sampling well enough. And I think, maybe that we need to bias more towards having not quite as good energies, but having better sampling. And, but I worry, because MCCE is not enough without MD to give us conformational changes, but every conformational trajectory is biased by the charge state.
Absolutely. Stephen?
Yeah. Hi, Marilyn. This was very interesting. This what you've described is sort of a nightmare scenario, and one that I've worried about for a long time.
It's my job.
Yeah, thank you for making us sleep better. Coming at this as an experimentalist, I guess the question is there a key change you could suggest in it? Let's say don't talk about this, complex one, it's much too complicated. Go back to lysozyme or something like that, where every mutant has been made, and really ask, well, if you make that change and now you worry about the problems you're worried about, did it really matter? And did it change in a way that you would have predicted? Or I mean, I was thinking perhaps just something like exchanging H for D. What would you predict? Because really the question here is, this sounds like a nightmare, but maybe it's not quite as bad as it seems.
I'm thinking back to your hole burning experiments. I mean, this is where bandwidth comes from. It's one of the things that underlies bandwidth.
Inhomogeneous broadness.
Inhomogeneous bandwidth. I mean, this is one of, everyone I'm an experimentalist in my DNA, and everything has a distribution, and the distribute, so it's not, delta functions. And so that's one thing, is that I think experimentalists should say, oh yeah, I always see a distribution of rates, or, whatever. I mean, it's why you don't titrate like as a step function. You titrate as a over 2 pH units. Now, what this means in terms of experimental, I am really keen, because I'm also working on my NSF renewal, how to think about that. I mean, I don't know whether the density of states or, whether we, how to prove, I mean, some of it's obvious. You get lysozyme, you get all the titration, you get the right PKs. So you know that there's a distribution of states underlying it. And you should have known that before, you just weren't paying attention to it. But I think it is interesting. I mean, you should see changes. We are thinking, I'm sorry, Steve, we are really thinking about looking at doing multiple changes, not doing a single change, because each change is correlated with other changes. So trying to find kind of nexi or groups where their protonation are correlated and changing both of them or changing a group of them and seeing whether we can do that. And I mean, we can make predictions in silico.
Yeah.
But I'm not sure what the measure, how the measurements are going to change yet.
No, I mean that's really what I'm getting at. And the question is this a nightmare for computer simulations that really doesn't impact experiments, or does it? I mean, that's it.
I think as an experimentalist, I'm going to say this is what experimentalists knew about already, which is that there's width, there's inhomogeneity. And I think, I was actually talking to somebody who was doing some basic stuff with NMR, and they wanted to get things so simple that they literally got rid of all the protonatable residues, because they said as soon as they had a single protonatable residue in there, they had distributions of protonation states. They didn't have a single protein.
Yeah.
And so I think what it should say to experimentalists is, yeah, this is what's going on. I also think that this is kind of why proteins chose asp, glue, arg, lyse, his, is that the asp and the glue and the arg and the lyse tend to be relatively quiet around physiological pH. Not every protein lives at pH 7, but the ones that do, it's kind of a flat region. And the his gives you the diversity of states. So at least you can do that if you want to do that. I mean, my proteins want the complexity because they want to have these metastable states they can move through.
Yeah. Thank you.
If you have any ideas of how to test it, you know where to find me. I'm happy to make any predictions you would like that you can disprove.
Jim?
2 questions, comments. Thank you. One, the more complex one, so you showed lysozyme and lots of charge states, but those charge states don't really care because they're on the surface, except for the asp glue, of course. Whereas in membrane proteins, and I might well have missed this, I'm presuming that if groups are more buried from solvent, they will be so tightly coupled that the net charge must be a tight constraint on what in my observation and in broad terms, the net charge must be a very tight constraint on what's going on. That's the first question. Second question is, I think it's asp, glue, his, lyse, arg, so all experimentalists can say, it's histidine, you don't need to do anything, folks. Sorry.
So one is, I think that the asp, glue, his, lyse, arg does kind of keep it a little quieter for protein. So this his, this is for lysozyme, and the asps, are flickering on and off. This is at pH 7, so even the lyse is flickering on and off, and so again, it's largely quiet except for the his, but not entirely quiet except for the his. And again, I think in a way you're right with the choosing, kind of having a buffer where it likes to be in a certain charge state. So this is a little busy, but these are all the microstates that have a charge of -3. And there are a lot of them. And this cluster never gets to -2. There's almost no way that it's going to lose, bind the third proton. It really likes to go between 1 proton and 2 protons. I mean, but again, that's something a protein can tune. I mean, it can say, oh, I'd really like to have 3 protonation states and move the pKs a little closer together, but it can also separate them. And I think that is often what happens. You don't need to be in a given protonation state. You can change it without anything bad happening in a lot of proteins, but there are kind of limits. That's
a priori you don't know.
A priori, you don't know. In fact, for this thing, people thought that it actually had a net charge of -5 with no protons in the basis state, and MCCE said, no, we think it's probably minus, with 1 proton bound. It's probably not with no protons bound. So that, that was kind of the people's intuition on it was wrong.
Okay.
Okay.
So I see no more questions. Oh no, yes, actually there is one question from Antonio.
Sorry again.
No.
It's just more a comment on your comment about our intuition. I think one of our problems dealing with this complex phenomena is precisely that our intuition is not designed to this kind of thing. What I mean is, our effort is try to understand the mechanistic aspects in a way that we understand, but maybe this is a completely unlocalized effect. Because from the viewpoint of evolution, I mean, something which is obvious for us is not necessarily more difficult, more easy to take place. So things are maybe completely delocalized. I think Steve's in his talk talked about that, the possibility that some effects are largely unlocalized or global or whatever you want to call them. And I think this is a clear example where we are dealing with this kind of thing. That's my view, at least.
I mean, we really feel like the network analysis for proton transfer paths really says you don't have to just look in the structure and go here to here to here. I mean, it's just, okay, if it's here to here to here, you see it. We see it in the network. But if it's like, sometimes it is just, okay, then actually one of the things we can do is we can see which are the low energy paths, which I didn't show, but we're beginning to do that. So not all paths are equally favorable. But I think, and again, the Monte Carlo really gives you access to complexity, and other methods don't. And if you don't have access to complexity, you don't see complexity. And people don't like complexity. I like to keep Steve up at night. That's been, motivating me for decades.
I think one of the problems is that, I mean, if you look at textbooks are full of very clear and specific mechanisms.
Right.
And that's, I mean, for me, that's a constant fight, trying to remodel the way people think about biological processes, because I don't know, I mean, but I'm philosophizing.
I mean, I think it's not just textbook, it's also, our colleagues,
Sure.
Who want to say, if I make this mutation, that's going to do it. And I mean, another thing that we see is in these really complex patterns, mutations do not normally knock out function. They 20%, this, that, but it's, or they're not super conserved in evolution. The simple patterns, that glue that's got to be there, you knock it out, you're dead. These other 6 residues that are doing this, people have knocked this out and that out. So, I mean, we really have to, we're really thinking about how do you prove something when it's just this complicated group and it's more robust, maybe it's more robust. And I don't know why why proteins use these more complicated motifs when there are simple ones.
More complicated for us.
More complicated for us.
I think the biology wants always an escape way.
Yeah,
exactly. This is the evolutionary strategy. I mean, you would never want a deep trap. You're trapped.
Yeah.
Yeah. So I think that's, it gives you a lot of robustness. And we see that in the evolutionary patterns. These complex things are just, I mean, we're now starting to say we're going to do covariant evolution and we're going to do multiple mutations, and we're really going to have to try to kill it. Where's the node that where's the cluster that we can kill it with?
Hmm. Okay.
Thank you.
Thank you, Marilyn. So I enjoyed very much the discussion of today. Next week we will have Antonio speech, and afterwards we will have a roundtable. Which, but I would say that, well, we have a roundtable after every talk, basically. So
Right.
not very different from the usual. Okay, so see you
All right,
next.
thank you, Walter. Thanks, everybody.
Thank you. Thanks, everybody.
Bye.